Okay. Um, yeah, titles kind of says it. Totally not me procrastinating. I mean, it's still the 9th of March. It's still the 9th of March, you know. Trust, bro. It's actually so ironic. Literally Sunday, which is supposed to be like the freest day I have, about about to be like the least amount of time I spend studying. Yikes. Yikes, yikes, yikes. Did I study ketogenesis yesterday? I think I did.
So much that it can be exhaled. Paint remover a problem in
Yeah, I
normal.
did. Okay. Protein catabolism. I'm gonna have to restart my computer. It's so laggy. I don't know if it's the computer or the website, but my laptop's been doing this. I want to buy a new laptop for my editing stuff. I think I'll be able to buy it in August with my internship money. That's exciting. This computer is like so slow for rendering projects. So slow. 2 minutes 45. Okay.
In this video, we'll be discussing protein catabolism. Proteins that you eat from your diet are broken down by proteases into the individual amino acids that form the protein. Those individual amino acids all have the structure like this. On the side, we have an amino group here. We've got our side chain. We have our acid group on this side. The nitrogen carbon-carbon is the backbone of the amino acid. This is the part that is the same from amino acid to amino acid. The side chain, the R group, is what's different from amino acid to amino acid. So those individual amino acids can be used to construct our own proteins if we want. Take them and build them into the molecules that our cells need to run, reactions and form structures and whatnot. Alternatively, down even further, you can remove the nitrogen group, the group, and you can remove the carbon skeleton here. So the amino group, the nitrogen there, can be used to create nitrogen-containing compounds. Things like our bases in DNA and RNA that contain nitrogen. Or the nitrogen can be processed into urea, which can be then eliminated a waste product via the kidneys. This is taking place in the liver and processing of the amino group. The carbon skeleton has two fates, depending on the side chain of the amino acid. Some amino acids are what we call glucogenic amino acids. This means that carbon skeleton can be used to generate glucose, so it can enter the gluconeogenic pathway. Other carbon skeletons, other amino acids, are said to be ketogenic, which means that their skeleton will be converted to acetyl-CoA and then ultimately to ketone bodies. There are actually only two amino acids that are strictly ketogenic, and that's lysine and leucine. There are some amino acids that are both ketogenic and
Lysines positively charged. Wait. Leucine is, I think, nonpolar. Okay, and then leucine. Yeah. It's nonpolar. Looks like valine, but with an extra. Yeah. Oh yeah, because isoleucine, right? Okay.
glucogenic. These are phenylalanine, isoleucine, tryptophan, tyrosine, and threonine. Everybody else, apart from those 7 that we just mentioned, are strictly glucogenic. Let's summarize what we talked about in this video.
Bro, do I have to
We
know
covered
that?
that's lysine and other carbon skeletons, other amino acids, are said to be ketogenic, which means that their skeleton will be converted to acetyl-CoA and then ultimately to ketone bodies. There are actually only 2 amino acids that are strictly ketogenic, and that's lysine and leucine. There are some amino acids that are both ketogenic and glucogenic. These are phenylalanine, isoleucine, tryptophan, tyrosine, and threonine. Everybody else, apart from those 7 that we just mentioned, are strictly glucogenic. Let's summarize what we talked about in this video. We covered the very basics of protein catabolism, what you can use the individual amino acids for, and what you can use the breakdown products of the amino acid for itself, the nitrogen group, the amino group, and the carbon skeleton.
Right. Ketogenesis. In fasting or low carbohydrate state, acetyl-CoA can be diverted to ketone bodies. Liver ketones can released in the bloodstream, can be used in ocean field storage by tissues in the brain, muscles, and heart. Precursor synthesis of cholesterol. Because it's fatty. Cholesterol is it's a steroid. Essential for synthesis of steroids, bile acids, and membranes. Yo, it is. It's true. Yo, tomorrow's Monday, bro. I mean, today is Monday. Ridiculous.
In this video, we'll be discussing a summary of the metabolic pathways. Our main metabolic pathway is glycolysis, where we take a little bit of glucose and we break it down into pyruvate. That pyruvate is then converted into acetyl-CoA, and the acetyl-CoA is put into the Krebs cycle. We generate a little bit of net ATP here in glycolysis. We generate a lot of reduced electron carriers, NADH, FADH2 here in the Krebs cycle. Those electron carriers can then be put to the electron transport chain to generate even more ATP for us. So this is our main metabolic pathway. This is, assume that we are in a fed state and we have supplies of glucose. If we have even more glucose than we can use at the moment, glycolysis is running to the max, Krebs cycle is running to its max, and we might choose to store that glucose in a couple of different ways. The first way would be to store it as a glucose polymer called glycogen. This would be a little glycogenesis. And this is literally a whole bunch of glucose molecules joined up together to form this big glycogen polymer. The other thing we can do is take some of these acetyl-CoAs that we're generating and shuffle them into fatty acid synthesis. This is not a direct storage form for glucose, but either
Please play, bro.
Either way, we could potentially break this down later to use as energy when we're not in a fed state. So let's say it's morning. You've had an overnight fast. You ate dinner last night, let's say around 7-ish, 8-ish. And it's now 6 o'clock in the morning, so you've gone about 10 hours or so without food. You're going to need to keep your blood sugar levels up. So the first thing your body is going to do, it's going to try and break down that glycogen through a little glycogenolysis to form our free glucose molecules. These can go off into the bloodstream and feed the other cells in your body. The second thing we might do is choose to make some new glucose from some of these glycolytic precursors. So for example, we can run a little gluconeogenesis. In gluconeogenesis, we're going to take pyruvate, convert it into oxaloacetate, and then, through a series of reactions, convert it back into glucose. So either glucose released from glycogen or gluconeogenesis is going to release free glucose molecules into the bloodstream that other cells can use for energy. If the starvation persists, maybe it's now been 24 hours since you ate, you didn't have time to eat, or maybe you are fasting for the day, or who knows. Then your body is going to switch to fat metabolism and start breaking down some of those fatty acids that you created earlier in times of plenty. So a little fatty acid oxidation, that's going to create our acetyl-CoA molecules again, which then go through the Krebs cycle and generate electron carriers. The problem with this pathway, though, is that if you are in a starved state, then some of this oxaloacetate is being drawn off into gluconeogenesis. And so the Krebs cycle might not be running as efficiently as it could. So some of these acetyl-CoAs are going to start building up. We can convert those acetyl-CoAs into ketone bodies through ketogenesis. Ketone bodies can easily travel in the bloodstream, and they can also pass through the blood-brain barrier, so they can enter the brain cells. Brain cells are unable to use anything except glucose or ketone bodies as fuel. In the brain cells, the ketone bodies can be reconverted into acetyl-CoA and used to run the Krebs cycle. So this is a means of feeding your brain, and like I said, an easy way for these things to travel through the bloodstream since they're fairly polar. The last thing that we can put on to our metabolic summary here is protein breakdown. We can do a little protein catabolism, and that will generate two types of amino acids for us, our glucogenic amino acids and ketogenic amino acids. Glucogenic acids are those where the carbon backbone can be used to generate glucose. It can go into pyruvate. They can actually enter in several different places along this gluconeogenic pathway. The ketogenic amino acids can be created into acetyl-CoA, converted into acetyl-CoA, which can then be used for ketogenesis or to run the Krebs cycle. So our black pathways here are the preferred pathways. That's the pathway that's running when you're in the well-fed state. The green pathways are the pathways that your body's first going to turn to when in a temporary state of starvation. And if that starvation prolongs, then we're going to switch to these red pathways, fatty acid breakdown and ketogenesis. Protein catabolism kind of comes in both places. We can start protein metabolism early because you have a ready supply of proteins in the blood already. Your plasma proteins, you've get free amino acids in the blood. But we don't want to do that for too long because protein breakdown is going to be detrimental to your overall body function. If you start breaking down your proteins, then you're going to become non-functional fairly quickly. So fatty acids are then the preferred source, and you'll burn fats until all of that is gone, and then you'll switch back to proteins. At that point, you're pretty much in dire straits. Let's summarize what we talked about in this video. We looked at a summary of the metabolic pathways, looking first at glycolysis and what happens in the fed state, including storage of glucose as glycogen and fatty acids. And then we looked at what happened in early starvation, glycogen breakdown, and gluconeogenesis, and later starvation, fatty acid oxidation, and ketogenesis.
I'm
And we looked
curious
at that
about
other cells can use for energy. If the starvation
glyconeogenesis and
persists, maybe it's now been
glycogenesis.
24 hours.
Was that in biochemistry 1? Glycolysis, glycogenesis.
In this video, we'll be discussing glycogen metabolism. Well, glycogen is basically a big storage form for glucose. So if you have plenty of glucose around, it makes sense to want to store as glycogen. And if you need glucose, then it makes sense to break down that glycogen molecule to release that glucose into the bloodstream. So the formation of glycogen is called glycogenesis. Genesis is formation of something, and the breakdown of glycogen is called the glycogenolysis. Lysis means to break something down. So when you have a lot of blood sugar around, the hormone released is insulin. And we already know insulin from our glycolysis pathway. We know that insulin stimulates glycolysis. And again, that makes sense. If you have a lot of blood sugar around, you'd like to try and break that down. However, if your glycolysis pathway is running to its max, and may even be tapped out a little bit, then it would make sense to take that glucose and put it into the storage form. So in glycogenesis, glucose monomers are going to be converted to that glycogen polymer, and that polymer is then stored primarily in the liver, and to some extent is stored in skeletal muscle. Now, The opposite situation, when you have low blood sugar levels, the hormone released there is going to be glucagon. And again, we already know glucagon from gluconeogenesis. Glucagon is the hormone that stimulates the formation of new glucose when glucose levels are low. So it makes sense that it would also be the hormone that stimulates glycogenolysis to break down that glycogen polymer, again, to get our blood sugar levels back to normal. So glycogenolysis is the breakdown of the glycogen polymer into glucose monomers. Now, the other hormone that will stimulate glycogenolysis is epinephrine. You might recognize
Takes it from the blood.
epinephrine as our fight or flight hormone. That's our stress hormone. And so it also makes sense that epinephrine would trigger glycogenolysis, because if you're in a stress situation, you want to keep your blood sugar levels high to make sure you can deal with whatever it is that's stressing you. And you've probably noticed that if you have a low blood sugar in between meals, let's say, sometimes you feel a little jittery, you feel a little shaky. And that's the result that epinephrine producing not only the glycogenolysis effect, but also triggering some of those other fight or flight effects that you're more common with. All right, let's take a look at the process. So the first step is going to be to phosphorylate glucose to glucose 6-phosphate. We already, again,
Oh no, it's
recognize
gonna
this
walk
from
me through the whole
glycolysis.
system now.
The enzyme that runs this step is hexokinase, and we want this to happen if we have high blood sugar
I think
levels.
at least this is
So we have this
hexokinase is
marked in
familiar.
green to remind you. Insulin, also in green on our last slide. And again, if glycolysis is running along just fine, then we don't need to continue proceeding through this pathway. Obviously, the rest of glycolysis is not here on this pathway, but you can imagine it here. So what we're going to do then is take this glucose 6-phosphate and divert it, put it into the glycogenesis pathway. And the first thing we're going to do is to take that 6-phosphate and convert it to a glucose 1-phosphate, shift the position of the phosphatase molecule onto the
What
1
did you
carbon,
say?
and the enzyme that runs the genesis pathway. And the first thing we're going to do is to take that 6-phosphate and convert it to a glucose 1-phosphate, shift the position of the phosphatase molecule onto the 1 carbon. And the enzyme that runs, that is called phosphoglucomutase, mutating the position of a phosphate on glucose, thus the glucomutase. And then in a couple of steps, we're going to take that phosphorylated glucose to glucose 1-phosphate and attach it to our glycogen polymer. The enzyme that primarily runs that is glycogen synthase, and we can see our new glucose added here to the polymer. Let's take a brief look at some energy. Of course, we know we're going to need an ATP here in the very first phosphorylation, but we also need a UTP that comes in here. And in fact, the glucose is attached to that uridine. We create uridine diphosphate glucose, UDP glucose, and that's the energy. The loss of that phosphate bond is going to drive this reaction forward. That's the energy used reaction. So we're first going to attach our glucose to the uridine, and then glycogen synthase removes the uridine and sticks the glucose onto the glycogen. The reverse pathway we're going to have in red, because we have glucagon and epinephrine there in red on the other side. So when your blood
Not gonna lie, a lot of this is going over my head.
sugar levels are falling, that primarily runs that is glycogen synthase, and we can see our new glucose added here to the polymer. Let's take a brief look at some energy. Of course, we know we're going to need an ATP here in the very first phosphorylation, but we also need a UTP that comes in here. And in fact, the glucose is attached to that uridine. We create uridine diphosphate glucose, UDP glucose, and that's the energy. The loss of that phosphate bond is going to drive this reaction forward. That's the energy used to drive the reaction. So we're first going to attach our glucose to the uridine, and then glycogen synthase removes the uridine and sticks the glucose onto the glycogen. The reverse pathway we're going to have in red, because we have glucagon and epinephrine there in red on the other side. So when your blood sugar levels are falling, then the first thing we want to do is to pop a glucose off of glycogen. So the enzyme that does that is called glycogen phosphorylase. And actually, what he does is phosphorylate this glucose. He phosphorylates it on 1' carbon and then pops that guy off of there. So here we have glucose 1-phosphate. Then we're going to convert that back into glucose 6-phosphate, again, using our phosphoglucomutase enzyme. Then our ultimate goal is to get that glucose out into the bloodstream. Remember, this is happening in the liver. The liver's the guy that stores glycogen and then also releases glucose into the bloodstream by breaking down glycogen. So we can't release this phosphorylated guy into the bloodstream. Glucose with a phosphate on it won't cross the cell membrane, so we have to dephosphorylate it. We're going to use our enzyme glucose 6-phosphatase that creates a non-phosphorylated glucose that we can now release into the bloodstream. Let's summarize what we just talked about. We discussed the basics of glycogen synthesis, called glycogenesis. We said this occurs when we have high blood sugar levels around and want to store some of that as polymer. and we discussed the basics of glycogenolysis. This was the breakdown of glycogen under low blood sugar conditions. This called glycogen. Then our ultimate, because we have glucagon and so we're first going to attach our glucose to the uridine, and then glycogen synthase remain here, and in fact attach it to our glycogen polymer. That is called phosphoglucomutase, mutating the position of a phosphate on glucose, thus the glucomutase. And then in a couple of steps, we're going to take that phosphorylated glucose to glucose 1-phosphate and attach it to our glycogen polymer. The enzyme that primarily runs that is glycogen synthase, and we can see our new glucose added here to the polymer. Let's take a brief look at some energy. Of course, we know we're going to need an ATP here in the very first phosphorylation, but we also need a UTP that comes in here. And in fact, the glucose is attached to that uridine. We create uridine diphosphate glucose, UDP glucose, and that's the energy. The loss of that phosphate bond is going to drive this
The
reaction
we're to
forward.
attach it to the actual
That's the
chain
energy
of glucose.
used to drive the reaction. So we're first going to attach our glucose to the uridine, and then glycogen synthase removes the uridine and sticks the glucose onto the glycogen. The reverse pathway we're going to have in red, because we have glucagon and epinephrine there in red on the other side. So when your blood sugar levels are falling, then the first thing we want to do is to pop a glucose off of glycogen. So the enzyme that does that is called glycogen phosphorylase.
We're adding
And
a phosphate
actually,
back.
what he does is phosphorylate this glucose. He phosphorylates it on 1' carbon and then pops that guy off of there. So here we have glucose 1-phosphate. Then we're going to convert that back into glucose 6-phosphate, again, using our phospho.
Yo, 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 yo. I wasn't done. I wasn't done. I wasn't done. Yo, yo, yo. I'll do this tomorrow. Not right now. Not right now.
This video, we'll be discussing the pentose phosphate pathway. The pentose phosphate pathway is also known as the hexose monophosphate shunt. A hexose is a 6-carbon sugar. Monophosphate means that there will be a phosphate attached to one of the carbons on that sugar. So, for example, glucose 6-phosphate is a hexose monophosphate. Shunt means that we can draw this product out of an existing pathway and shunt it through a completely different pathway to generate some different products. Ultimately, we can create some products that could go back into the original pathway. So we are shunting glucose 6-phosphate out of glycolysis, putting it through this pentose phosphate pathway, and can ultimately shuttle it back into glycolysis if we need to. There are 2 phases to the pentose phosphate pathway. The first phase is called the oxidative phase, because we're going to be doing some oxidizing of glucose 6-phosphate. So glucose 6-phosphate is oxidized to ribulose 5-phosphate. This would be an oxidative decarboxylation. The enzyme that runs this phase is called glucose 6-phosphate dehydrogenase, or just G6-PDH. Now, because something is getting oxidized, something else is going to have to be reduced. And the molecule that's getting reduced in this case is NADP. We're going to reduce 2 molecules of NADP.
We use this in, we use NADPH in the fatty acid break. Ketogenesis. Yeah, we use this in ketogenesis, fatty acid breakdown. Please play.
To produce 2 molecules of NADPH. There are several steps here, but this is the overall reaction. G6-PDH will oxidize glucose 6-phosphate to ribulose 5-phosphate, producing 2 molecules of NADPH in the process. The NADPH is the regulator for this process. Now, glucose 6 phosphate dehydrogenase is inhibited by high levels of NADPH. And that makes sense because if you have a lot of NADPH around, there's no point in running the reaction to produce more NADPH. So you might as well use that to inhibit the enzyme. The second stage of the pentose phosphate pathway is known as the non-oxidative phase. And this is basically a series of reversible rearrangements of the carbon chain to generate some glycolytic intermediates, as well as ribose 5-phosphate, which we can use for nucleotide synthesis. So the first step, we can convert this ribulose 5-phosphate into a ribose 5-phosphate. This can be drawn off into, like I said, nucleotide synthesis. Or we can combine these two 5-carbon guys to essentially create our ribose 5-phosphate and 5-phosphate to essentially create a 10-carbon molecule. And we can split that apart into a 3-carbon molecule and a 7-carbon molecule. Now, the 3-carbon molecule you'll recognize as glyceraldehyde phosphate. This is one of the glycolytic intermediates that could be produced in this phase of the pentose phosphate pathway. We're going to indicate these all with a green box and a little green star there. So glyceraldehyde phosphate is a glycolytic intermediate. The 7-carbon guy, we don't need to worry about. But we could also take these 10 carbons and split them differently to produce fructose 6-phosphate, a 6-carbon guy, and a 4-carbon guy. The fructose 6-phosphate, again, you will recognize as a glycolytic intermediate. The 4-carbon guy, again, we don't have to worry about. And lastly, we can take our 4-carbon guy, combine it with yet one more molecule of ribulose 5-phosphate to create a 9-carbon structure that we can split apart into a 6-carbon guy and a 3-carbon guy. 6-carbon, again, are fructose 6-phosphate, and the 3-carbon are glyceraldehyde phosphate. So these products can be shunted back into glycolysis. Now, the advantage of this is that it gives us some variability in this pathway. Let's say that the cell absolutely needs NADPH, but does not require any ribose 5-phosphate. So it could run the first part of this pathway. All of the ribulose 5-phosphate could get rearranged into the glycolytic intermediates, and these can all go back into glycolysis. Or let's say we need both NADPH and ribose 5-phosphate. Then we're going to run this pathway. Some of this ribose 5-phosphate will be converted to ribose 5-phosphate to go into nucleotide synthesis, and the remainder can be reconverted into glycolytic intermediates. Or what if we only needed ribose 5-phosphate? Let's say there was plenty of NADPH around, and this glycolysis. Now, the advantage of this is that it gives us some variability in this pathway 5-phosphate. There are various number of anabolic reactions. For example, fatty acid synthesis relies very heavily on NADPH for
Bro, it's synthesis? Oh,
reducing power. Also, it can act to help eliminate reactive oxygen species. So there's always some reactive oxygen species, roses, formed during cellular metabolism, and NADPH produces reducing power in order to eliminate those guys. This is particular because they can only rely on the pentose phosphate pathway for their NADPH needs. Other cells can generate through other pathways, but red blood cells rely exclusively on pentose phosphate pathway for the NADPH. So people who have a deficiency in NADPH, maybe due to a lack of that particular enzyme, a genetic G6PD-H deficiency, have the inability to generate NADPH, and it often leads to anemia, because the oxidative damage to the red blood cells causes the loss of those red blood cells and anemia. Ribose 5-phosphate, we mentioned, was used for nucleotide synthesis, and that's pretty much it. Here's a picture of ribose 5-phosphate. You'll recognize this as the sugar phosphate part of RNA for sure. And if we didn't have the hydroxyl group here, this is the backbone for DNA as well. Let's summarize what we talked about. We discussed the specifics of the oxidative and non-enzyme was being inhibited, then we can't really run the oxidative phase. We could take these glycolytic intermediates and work backwards from glycolysis up to ribose 5-phosphate that we could then use to generate nucleotides. And we could do this even if we weren't running the oxidative phase of the pathway. It just gives us some variability in the pathway. Let's talk a little bit more about the functions of these products here. So discussing the pentose phosphate pathway. The pentose phosphate pathway is also known as the hexose monophosphate shunt. A hexose is a 6-carbon sugar. Monophosphate means that there will be a phosphate attached to one of the carbons on that sugar. So, for example, glucose 6-phosphate is a hexose monophosphate. Shunt means that we can draw this product out of an existing pathway and shunt it through a completely different pathway to generate some different products. Ultimately, we can create some products that could go back into the original pathway. So we are shunting glucose 6-phosphate out of glycolysis, putting it through this pentose phosphate pathway, and can ultimately shuttle it back into glycolysis if we need to. There are 2 phases to the pentose phosphate pathway. The first phase is called the oxidative phase because we're going to be doing some oxidizing of glucose 6-phosphate. So glucose 6 phosphate is oxidized to ribulose 5 phosphate. This would be an oxidative decarboxylation. The enzyme that runs this phase is called glucose 6 phosphate dehydrogenase, or just G6-PDH. Now, because something is getting oxidized, something else is going to have to be reduced. And the molecule that's getting reduced in this case is NADP. We're going to reduce 2 molecules of NADP to produce 2 molecules of NADPH. There are several steps here, but this is the overall reaction. G6-PDH will oxidize glucose 6 phosphate to ribulose 5 phosphate, producing 2 molecules of NADPH in the process. The NADPH is the regulator for this process. Now, glucose 6 phosphate dehydrogenase is inhibited by high levels of NADPH. And that makes sense because if you have a lot of NADPH around, there's no point in running the reaction to produce more NADPH. So you might as well use that to inhibit the enzyme. The second stage of the pentose phosphate pathway is known as the non-oxidative phase. And this is basically a series of reversible rearrangements of the carbon chain to generate some glycolytic intermediates, as well as ribose 5-phosphate, which we can use for nucleotide synthesis. So the first step, we can convert this ribulose 5-phosphate into a ribose
Indirectly.
5-phosphate. This can be drawn off into, like I said, nucleotide synthesis. Or we can combine these two 5-carbon guys to essentially create our ribose 5-phosphate and another ribulose 5-phosphate to essentially create a 10-carbon molecule. And we can split that apart into a 3-carbon molecule and a 7-carbon molecule. Now, the 3-carbon molecule you'll recognize as glyceraldehyde phosphate. This is one of the glycolytic intermediates that could be produced in this phase of the pentose phosphate pathway.
I'm gonna worry about this tomorrow. I love you. Bye.